Good afternoon. To those here at this very special event today celebrating Gerald Westheimer's centenary, and to Gerald's many friends, colleagues, and admirers around the world joining us virtually, I understand there are more than 50 right now. Welcome to this very special program, Optometry and Vision Science, the Next 100 Years, which features 3 talks, all curated by Gerald. We all know Gerald, but for those who don't really know Gerald, he is one of a kind. He's a giant in the field of optics and vision. He's a pioneer in understanding the factors that limit normal vision from optics to perception. For many of us here, whether in person or virtually, he's also a colleague, a friend, and a mentor. For those who don't know about Gerald's journey to this point, I want to read you Suzanne McKee's loving introduction to the Festschrift that honored Gerald on his 65th birthday. Gerald's career did not follow the conventional path to academic achievement. In fact, as he is fond of telling his friends, he never graduated from high school. In 1938, the Westheimer family had fled from the terror of Nazi Germany to a strange and beautiful country halfway around the world. In Australia, diminished circumstances forced the teenage boys to find jobs, learn English, acquire a trade. Gerald was apprenticed to an optometrist and took courses at night to learn the requisite skills for optometric practice. He still takes pride in the clinical skills learned as an adolescent and maintains his license as a registered optometrist in the state of New South Wales in case, as he slyly remarks to academic colleagues, he should ever have to earn a living. In another respect, this trade was an excellent choice because it encouraged Gerald's nascent interest in optics. After receiving a Bachelor of Science degree in mathematics and physiology through the arduous course of evening classes and part-time study, Gerald was awarded a fellowship of Sydney Technical College for his thesis on the optical theory of contact lenses. In his own remarkable autobiography, Gerald wrote, "As an optometry student in the early 1940s, I wondered whether the dogmas based on vague clinical experience that constituted much of our training in those early days of the profession might not find more solid substantiation in science. If uncertainty clouded the diagnosis based on patients' recital of their symptoms and on subjective tests, or the prognosis and therapy of myopia progression, surely this was because we were only at the beginning of the scientific exploration of diagnosis and treatment of eye conditions." And as the record shows, Gerald, for more than 75 years, from his earliest publications on the optics of contact lenses in 1948 and early 1950s, has continued to publish on that topic as recently as 2023 in this very same journal as his first paper. Indeed, Gerald made seminal discoveries in understanding the optics of the eyes, binocular vision, spatial vision, eye movements, learning, and visual illusions. When Gerald was elected, for Gerald's election to the Royal Society in 1985, Horace Barlow wrote, "He has made diverse and important discoveries about the visual system over the past 30 years. These include, 1, the mathematical analysis of disjunctive eye movements as a control system, an analysis with the greatest predictive power so far achieved on a biological system of this type. 2, the objective determination of the quality of the retinal image. 3, the influence of the cerebellum on eye movements in primates. 4, the mechanisms for achieving high positional acuity in vision, hyperacuity, and the reason for the small effect of image motions on this ability. His work is respected for its reliability, diversity, and the depth of its analysis. He is internationally regarded as the leading exponent of physical methods in the analysis of vision." As Barlow noted, one example of Gerald's many contributions, one which is closest to my own heart, is his work on spatial vision. He showed that humans are able to discern small changes in the relative position of a stimulus that are an order of magnitude smaller than the smallest foveal cones in the retina, even when the stimuli are moving over many photoreceptors. He termed this remarkable ability hyperacuity. a term that is still widely used and elucidated many of its properties. This work inspired a whole new field of endeavor aimed at understanding this remarkable visual capacity. But Gerald did not stop there, and he's continued to make important contributions, publishing high-quality, important papers every year, up to and including his most recent essay on Gestalt theory, published in the Journal of Vision in 2023. Gerald's many seminal discoveries in understanding the optics of the eye, binocular vision, spatial vision, eye movements, perceptual learning, and visual illusions have been widely recognized. He's a Fellow of the Royal Society and a Fellow of the American Academy of Arts and Sciences. He's received prestigious awards, including the von Selman International Prize in Vision and Ophthalmology, the Glenn A. Fry Award from the American Academy of Optometry, the Prentice Medal from the Association for Research in Vision and Ophthalmology, and the Order of Australia, to name just a few. I think Tony Movshan actually put his finger on a key reason for Gerald's success. To quote Tony, "Gerald loves knowledge. For him, the joy of acquiring a new understanding is boundless and endlessly motivating. It can be an understanding about physics or mathematics or music or literature or how vision works." And I think everyone who knows Gerald will wholeheartedly agree. Please join me in wishing Gerald a happy, healthy 100th birthday and many more. Gerald, you are an inspiration to us all.
Thank you, Dennis. Thank you all very much for coming. It's been a long road. And it is not automatic. When you consider that what happened to boys born in Berlin in 1924, depending on their ethnicity, they were either ground up by the war or by the Holocaust. So very few of us were fortunate enough to have parents who understood what is going to happen and got out and saved our lives. So that I'm here is entirely a tribute to my parents for their foresight. What is happening here today also, the theme of today's event has also a long history. It goes back 80 years. When I was an optometry student in Sydney, starting seeing patients, being in the eye clinic, and recognizing that there was something uncertain about what we were doing. As my friend Archie Polsky used to say, well, what is clinical experience? To him, clinical experience is when you make the same mistake 10,000 times. So it was a very difficult period and worth remembering what it was like. The war was grinding on with uncertain outcome. Society was uncivil, there was prejudice, there was oppression, and when you go into social activities, there was irrational, things were not rational. And when you go in the clinic, things were uncertain. So all we had at that time, we're talking 80 years ago, all we had is a future. We had a hope that the future would be different. The future would be different in interaction between people, in society, more rational, and when we go into practice, high practice, and science, it would be more certain. You would be knowing what you would be doing. So there was fantastic hope that drove us forward, and there was an immense opportunity. We are, what it opened up then, and I was extremely fortunate to participate in, was the second half of the 20th century, with science in America. We could do things that could not be done before. We had new light sources, the laser came, we had electronic devices, we had computers. Later on, we had the modern biological techniques. So we had an opportunity to do research, to find things out, to have developments that the previous generation did not have. And I think we did the best we could doing that. Now, fast forward to the end of the 20th century, we went through this while there was a semicolon. Things were not the same. And the reason why they're not the same is what used to be a tenet of dialectical materialism, which we studied and wasted our time on. But one tenet was Hegel's phrase that a quantitative change in sufficient measure becomes a qualitative change. So when you have scientists increasing in numbers by a factor of 10 or 100, and the techniques allowing things to do by a factor of 10, 100, a billion in computers, things are not the same. And science now, I think during the last 20 years, is in a different configuration. Where you have many people needed to do something. Many more facilities are needed to be brought to bear on a problem. And that changes the personality, not only in the number of scientists, but the quality of scientists. What we had to deal with as young scientists was the harsh judgment of people like Rushton and Burnley, who you made the slightest mistake in logic, you were dumped on. You couldn't, you were afraid of it. You did your own work, and one or two people, almost no papers had more than 2 authors. All of this has changed now. And a different kind of approach is needed to do science. So things will now be done in a way that we couldn't do. And that's what we are looking forward to. So the theme now is not what happened last century, which may be forgotten, but maybe shouldn't be, but what will happen in the next century. So that's today's theme. What will the eye care professions look like in the next 100 years? What are the techniques? And so we're looking forward to our talks today, because this is where the future lies. Thank you for coming. Thank you very much for everything.
Thank you, Gerald. It is such a thrill and an honor to be part of this celebration. I hope you all agree. And it was not too difficult to plan. Marla and I met Gerald in September. And he baked the cookies, he prepared the tea for the meeting, and he said, this is what the program has to be. He literally gave the titles for the program on that day. And that just set the wheels in motion that led to the program that we have today. So thanks, Gerald, for your foresight. And the only contribution I had was, let's call it Optometry and Vision Science the next 100 years. That's my small contribution. Also, thanks to the staff from the optometry group, Matthew and Caitlin, who have put together all the AV and the live streaming. So thank you very much. So my job is to introduce the three speakers. Gerald already told you what the topics for the three speakers are. And I'll just tell you a little bit about the speakers. So the first speaker today is Aaron Lee. He's an associate professor and vitreoretinal surgeon at the University of Washington, Department of Ophthalmology, and the recent recipient of the C. Dan and Irene Hunter Endowed Professorship. He completed his undergraduate at Harvard University and medical training at Washington University in St. Louis. He chairs the American Academy of Ophthalmology Information Technology Steering Committee. He currently serves as an associate editor for both Translational Vision Science and Technology and Ophthalmology Science, and on the editorial board for American Journal of Ophthalmology and Nature Scientific Reports. He's published over 200 peer-reviewed manuscripts and is a leader in the field of artificial intelligence in ophthalmology, which we'll be focusing on today. His research is focused on the translation of novel computational techniques in machine learning to uncover new disease associations and mechanisms from routine clinical data, including electronic health records and imaging. And I keep asking him if he's nervous, but if he's not nervous about giving his talk today, he must be nervous about his position in the field, because every time I hear our current NEI director, Michael Chan, give a talk, he is always suggesting that Aaron and his partner, Cecilia Lee, are going to solve the world's vision problems. Okay, so no pressure. The title of Aaron's talk today is Artificial Intelligence in Eye Care Practice. Aaron.
So thank you so much for having me. I have to say this is the first time I've given a scientific talk at a birthday bash. So these are my financial disclosures. So I just want to start by giving this talk about AI to kind of level set where we are in terms of human society. So I think right now we are in the middle of what people are calling the fourth industrial revolution. AI and machine learning algorithms are powering everything that we see and do whenever we interoperate with an electronic device today. And these fields, these terms have formal definitions. It's good to know these because the media tends to use them interchangeably. But the field of AI is actually very, very old. It's been around since the dawn of modern computing. And it really is, there's a subfield within AI known as machine learning. And then there's a recently new field called deep learning. And so these are subsets of one another. So not all AI is deep learning, even though the media tends to use them interchangeably. To understand what deep learning is, you have to kind of understand how the original artificial neural networks worked. They were these fully connected layers called multi-layer perceptron networks. And they were patterned after what computer scientists understood how biologic neurons worked. And it was very challenging in the 90s to scale these to do anything useful. And so for a long time, these came and went, and the field of machine learning never really changed. There's sort of four advances that occurred around the same time that led to the birth of what we call deep learning today. The first was the realization that graphics cards could be used as linear algebra computing engines. The next was using a special filter called the convolutional neural network that was actually patterned after the visual cortex and the way that the brain and the retina processes visual information. The next was a nonlinear activation functions, and then finally our computing architecture had evolved to a point where we could amass large amounts of data and collate them. And that all led to this exciting time that we live in today. So for the last 5 to 10 years or so, it's hard to open the news without seeing something about AI doing something that as well as a human being, if not better. And this underlying technology is what gave birth to things like ChatGPT today. So just to kind of recount how this affected the field of vision science. This was one of our first works in the field. We wanted to see if deep learning could distinguish OCT scans from a normal versus AMD. And to do that, we took a data set of about 100,000 B-scan images, and we trained a state-of-the-art model at the time called VGG16. And within a day, we got these receiver operator characteristics curves. And I was shocked when the first model performance was kind of read out from the machine. I couldn't believe these results. They seemed too good to be true. All we were giving these machines, the algorithms, was just a whole bunch of images of normal and a whole bunch of images from macular degeneration, and it learned to classify these two classes on its own. In order to kind of make sure that the model was doing something useful, we did these post-hoc visualization tests to probe where the model was most dependent upon, and shown that the model was actually indeed looking at areas of pathology. And so that pattern of that template of describing the data set, showing the model schematic, showing the receiver operator characteristics, and these post-hoc visualizations became the template for many, many papers in the field. And that led us and many others to publish many, many papers showing that throughout the entire sort of spectrum of eye care, these deep learning models could actually impact the way that we deliver care. One sort of concrete example of this, in the US, there's 3 algorithms right now that have passed FDA approval. And in the field of eye care, we somehow went from like no AI to level 5 fully autonomous AI, with not a lot of AI in between. And so right now, these 3 algorithms, they are approved for diabetic retinopathy screening. So they can look at images, give a recommendation about whether that person should see an eye care provider. And that's all done without a human being in the loop at all. So we did a study to compare these models against VA teleretinal grader. And we invited a whole bunch of different companies. Some of these were FDA approved at the time. And ultimately 5 agreed. And we showed that in a large retrospective data set from the VA of more than 100,000 images, that these models had very high negative predictive value and unfortunately low positive predictive value. When we looked at its ability to screen for the higher levels of diabetic retinopathy, we showed that these models actually did as well as a VA teleretinal grader. It's hard to beat the VA teleretinal grader because they had 100% sensitivity. So the teleretinal grading system at the VA is actually working quite well. But this does hopefully lead to a future where these models can be deployed at population level for screening for diabetic retinopathy. Another sort of vignette that I want to kind of go over is the work that we did with the MACTEL Consortium, where hopefully we're going to step away from our sort of human grading systems that we've created in our field. So, human diseases don't follow discrete steps. They're not step functions. They follow some sort of continuous spectrum, and we need to understand how to build that spectrum. One of the things that in MACTEL Type 2, the traditional staging system is from Gaspoloti, has about 5 stages. More recently, Emily Chu created a 7-step scale based on visual acuity. And what we did is we combined 2 concepts from machine learning. One is this multi-view idea where we said each of the OCT B scans in a volume could be considered a different view. And then we used a dimension reduction technique that could reduce the OCT's image high dimensional information down to a single point on the 2-dimensional scale. And when we did that by training a deep learning model, we arrived at this manifold. So here, these dots are colored post hoc by the 7-step scale from Emily Chu. And you can see that they go from very normal in the upper left to very diseased in the lower right. Here's a video sort of fly through where we show the different OCTs at the different scales as you kind of go through the manifold. And you can see they start out very normal, and by the time you get down to the lower right, they're very diseased. So this is an example of how we can use these techniques and these AI models to transform the way that we understand human diseases today. A lot more detail about how we did the clinical validation of this scale can be found in this paper. I do want to spend a little bit of time talking about an important project that we're working on now that I think will hopefully affect this field for the next 25 to 50 years or so. One of the big problems with deep learning and AI is that they rely very heavily on the data sets they're trained with. And if there's biases that are built into those data sets, then the models really don't do that much better than those biases. One really important data set that people have been using for the last 10, 15 years is the UK Biobank. It's a really amazing project that's generated about 15 petabytes of data and many high-profile publications. But there's a critical problem with the UK Biobank in that diseased people are very uncommon in the data set, and it's 95% British white. So the average UK Biobank participant lives about 10 years longer than the same population they're drawn from. So everything that we've learned from the UK Biobank is really about extremely healthy white British people. The NIH started 2 programs in the US to try and build the flagship data sets for AI in medicine. One is the All of Us program, and that's patterned very similarly to the UK Biobank. The second is this Bridge to AI initiative. Cecilia and I put our project together, and we were lucky to be one of the 4 data generation projects that were funded. Our project is called AI Ready and Equitable Atlas for Diabetes Insights, and the overall goal of this is to build a data set that could be used to study a concept called salutogenesis. If you've never heard of the word salutogenesis, it's really just the opposite of pathogenesis. It's the way that people can go back to a healthy state after being in a disease state. So we wanted to build a data set that could be used to study salutogenesis in type 2 diabetes. We have a very extensive protocol that we are trying to recruit about 4,000 people into our study, where we do a whole battery of different tests, including many different surveys. We obtain many different forms of retinal imaging and do other things like a 12-lead EKG. We send them home with a fitness tracker and a continuous glucose monitoring device, and we built a custom environmental sensor that can read the pollution levels inside of their homes. And the overall idea here is that we're going to build a very large cross-sectional data set that at the end of the study will be balanced for 3 things. So it'll be balanced for disease. So we're going to, out of the 4,000, 1,000 will be normal, 1,000 pre-diabetic, 1,000 oral medication control, and 1,000 insulin dependent. Within that, there'll be a balance for race, so 1,000 white, 1,000 black, 1,000 Asian American, and 1,000 Hispanic. And within that, 1-to-1 balancing of male and females. So that's the idea. We have done a lot of work to try and make this data set easy for people to use so that they can start to do their AI work right away. And at the end of the day, we want to use that idea of that dimension reduction to build these manifolds of human beings that fall along this disease spectrum. And we can start to understand these more complex ideas like salutogenesis. So we are very close. Actually, in next week, we're going to have an open house at the NIH where we do the first public preview of this data set. And then 2 weeks after that, we're going to release it to the world. And anybody in this room can go and download this data set onto your hard drives to use for research right away. We are actually not allowed to do research on our own data until we release it to the world. We have a lot of opportunities for engagement. If you're interested in learning more about this data set, this is our website and our QR code. I encourage you to kind of follow along. And Michael Chang was mentioned. He's known for many things, but he's an informatician. And he's actually famous for developing some of the first AI models for retinopathy of prematurity screening. And so him being the director of the National Eye Institute, I think he's going to hopefully set a scientific program where data science is going to play a foundational role in everything that we do in the future. And, you know, I think 100 years, 50 to 100 years from now, it's going to be hard to imagine a time when AI was not part of a foundational thing that you're doing, when either it's science or taking care of patients. So it's very exciting times that we live in, and I just want to end by thanking all the team members in my lab and the funding sources. Thank you.
So we don't have time for questions because we want to get to the social part of the program, but there's one person who's always allowed to ask a question. And so, Gerald, if you ever have a question to ask, please don't hesitate. You do. Hand him the microphone. Sorry, hold on a second. We'll get the...
What do you think will be the role of AI in clinical practice 20, 30 years from now?
Yeah.
Or even 10 years from now?
Right. So I think that the way that AI is going to be placed in a pivotal role inside of the clinic, because it's going to start to transform the way that we interact with patients. I think the way that we, as eye care providers, we are forced to deal with many different things coming at us at the same time. There's all the imaging that we have to look at. We have to look at the patient. We have to listen to their story. But our attention has always been divided. And I'm hoping that AI will play a role in making some of those things kind of go away, and maybe highlighting things that we should pay attention to, and augmenting the way that we deliver care so that we can spend more time actually talking to our patient and taking care of them. I think it's going to change the way that we think about diseases and the way that we interpret all the imaging and all the clinical information that we have at the point of care.
Thanks, Aaron. Okay, next up. Next up is Susanna Marcos. She's the David R. Williams, who also happens to be sitting beside her, Director of the Center for Visual Science, Nicholas George Professor of Optics at the Institute of Optics and Professor of Ophthalmology at the Flom Eye Institute at the University of Rochester in New York. She started her academic career in Spain, getting her PhD in physics from the University of Salamanca, quickly rising up in the field and finally rising to the directorship of the Institute of Optics at the National Research Council in Spain in Madrid. Her work has been recognized with multiple awards, including the Adolf Lamm Medal and the Edwin Land Medal from Optica, or formerly Optical Society of America, the ICO Prize from the International Commission for Optics, the Ramon E. Cajal Medal by the Royal Academy of Sciences, and a National Research Award in Engineering by the Spanish government, the Jaime I Award, last two presented by the King of Spain. Yes, there is a King of Spain. She's a leading researcher in visual optics, having pioneered multiple technologies of eye, optical imaging, diagnostics, and treatments, including novel IOL designs. She's published more than 200 highly cited publications, supervised 24 PhD theses, co-inventor of 26 patents, and participated in 2 spin-out companies. From a personal standpoint, I never resist the chance to say this. She's my academic twin sister. We both gave our very first ARVO talks back-to-back in 1994, and have had surprisingly parallel career trajectories ever since. But after giving this intro, I think she's lapped me a couple of times now. So, Susanna.
Thank you, Austin. Thank you very much for the invitation. It's really a pleasure and an honor to be here. Gerald Westheimer, you've been my mentor, my inspiring person throughout the years. Your papers were always on my desk during my graduate studies. And also, every time I came to Berkeley, really, the conversations with you were the highlight. So I'm really honored to be here. It's actually challenging, and it's probably one of the most difficult talks I think I've ever gave, to think how the field is going to be 100 years from now. But also, it's been fun to prepare this talk. First, because it's good to reflect on how many things can change over the course of a century. Also, how what we do may be impacting the field in the next years. And also, play a little bit as a fortune teller and see how things are going to evolve and how they will look like 100 years from now. And actually, people imagined the world 100 years ago, and they came with this type of illustrations by French artist Villemart. Well, he was thinking that there would be machines that would convert knowledge and actually transmit the knowledge directly on the children's brains. So maybe Neuralink is doing that, or will be doing that next. He wasn't very accurate on the clothing fashion for the kids, but maybe happening soon. There is also, these ideas of that mailmen will be, like delivering letters by plane. Probably not very different from drones delivering packages by Amazon. There were also more futuristic views of Manhattan with these flying cars, and when we actually ask AI, and this is delivered by Midjourney, an AI illustrator, how the world is going to look 100 years from now. Well, you see these topic images of robots walking in the streets of very gloomy cities, and this blending of machines and people. You also see all these things that make reminiscence of virtual reality devices. And all this imagery probably comes from literature. And I'm bringing up here, just to put things in context on how the world is going to probably look like in 100 years. These ideas from an author, Pernar, of a book, Curious Matrix, where he thinks that global demographics is going to peak about 2080 and be 10 billion people in 100 years. The population will be mostly concentrated in cities, like mega cities and smart cities with new forms of transportation. It's interesting because some of the most prevalent conditions in vision, like myopia, is as related to urban populations. Also, in environmental changes like climate change that will actually produce large differences in the way the humans and the environment interact, and also change food supply and biodiversity. Major socioeconomic dynamics. Gerald was talking about how the world was during the World War. Major economic shifts are expected. Workforce changes, mainly driven by robots and AI. Social monitoring, also major differences in education and changes in geopolitical forces. Going closer to what matters today, technological advances. We've heard about AI, that it's likely revolutionizing all industries. It will be quantum computing, clean, limitless energy, virtual reality, lots of brain and computer interfaces, health and medicine, again, AI at the centerpiece in diagnostics and treatment, gene editing, robots, nanotechnology, increased lifespan, personalized medicine, regenerative medicine, 3D printing for affordable devices and prosthetics, telemedicine, and robotic surgery. And I think many of those can really apply to our field in optics and vision. It's interesting just for a couple of minutes to reflect of how things have changed over the century in our field. So when Gerald was born in Germany in 1924, if we look at optical societies, they were just started. The American Optical Association, then named American Optometric Association, was founded in 1898. By 1924, it has the first group of board of trustees. Optica had been around for a few years. In 1922, it held the first exhibit of optical instrumentations. And in 1924, the year Gerald was born, the Optical Society of America published the first translation of Helmholtz's book, three volumes of physiological optics, which I think he was really the influencer of the century. 1923, the German Society for Applied Optics was founded in Berlin. And then in terms of optometry education, you will know that there were already a few programs going on, some of them including their very own School of Optometry in Berkeley, being over a century old. So optical societies were there, and it's probably that in the next 100 years, we will have those, probably they will change and encompassing the changes in the world of bringing in other countries, particularly Asia. Optics industry, well, it's remarkable that many industry in optics was already founded in the mid-1800s. Around Rochester, New York, Kodak and Bausch and Lomb, also Corning, Massachusetts American Optical, which is the first optics industry in the US, Essilor, and then a few companies size Leica and Rodenstock in Germany. Remarkable that these companies are still in very good health today. So it will be probably likely that they will be increasing their area of operation and their portfolios, but probably still going 100 years from now. When you look at an optometry practice in the 1920s, and this is actually an illustration by Norman Rockwell that was commissioned by American Optical in 1927. Well, some things have changed, but it doesn't look that different, right? So the optometrist there is using straight retinoscopy. There is a visual field testing device, no computers, but doesn't look that different. But if you think about only a few years earlier, the eye examination was very crude. And it was in the 1920s where Gould-Strand slit lamp was commercialized. And this is the first images published by Alfred Bloch using the Gould-Strand slit lamp. Contact lenses, and Gerald Westheimer's PhD was on contact lenses, but they were only commercialized in the 1920s, although they had been conceptualized by Leonardo da Vinci in the 1500s. But they were probably not very comfortable. Blown glass, ground glass, commercialized by Zeiss and Miller. And as far as the intraocular lenses go, no intraocular lenses. They actually were invented a few years later. They were conceptualized in the 1600s by Daza de Valdez. And Claude Monet actually had his cataract surgery only 1 year before Gerald was born. Probably a very famous cataract patient because the changes in his art, but it was aphakic, no intraocular lens implanted in the eye. So you go now, well, the optometry or ophthalmology examination lane has some of the earlier instruments, of course, a bunch of other technologies, including corneal topography systems, many systems that include OCT technology that I believe is kind of the revolution of this century in ophthalmology, wavefront aberrometry. And I think there's still a lot to come. There is a number of technologies in the lab, and this is probably self-serving. I only put some photographs of my own lab, but we have adaptive optics, second harmonic generation microscopy, we have eye elastography. All these technologies, I think, still have the possibility to go and be converted into instruments that are used in the daily practice. Some of them, and again, these are examples from my own lab, are coming into the clinical practice, some in a small footprint, like wavefront aberrometry-based autorefractors or visual simulators. And there is actually much more of this technology to come. We're seeing many portable devices, fundus cameras, OCT systems, some of them point of care, that combined with telemedicine and artificial intelligence, as we've heard before, will allow for remote and automated diagnostics. So how is diagnostics going to be in 100 years? Well, it's hard to predict, but I envision kind of a very portable, maybe looking like current AR, VR systems that will encompass a number of technologies for eye imaging and including refraction, corneal microscopy, 3D biometry, fundus imaging, biomechanical properties of the eye, optoretinography, and of course, visual testing and visual simulation, which again, in combination with AI, will produce physical and functional biomarkers. These could be combined with lifestyle monitoring. Now we have these watches, but probably much more sophisticated 100 years from now, that in combination with molecular biomarkers, and of course, thinking on the context of the new computers that will be enabled by by new computers with different interfaces, much faster communications, perhaps quantum computers that can give you a very rapid multidimensional diagnostics. So what about visual aids? So what we have right now are spectacles, and well, they haven't changed that much for centuries. That's a painting by a Greco, so same type of eyeglasses than now. Contact lenses, well, they have developed thanks particularly to new materials. They come in monofocal or multifocals. Many of them are responsible. Intraocular lenses, there's 160 companies manufacturing intraocular lenses in the world. They come in different flavors, monofocal, multifocal, extended depth of focus, they're phakic or aphakic, also different materials, they're molded or lathed. So this is what we have right now. What I think is coming, well, I think these technologies, these visual aids are going to be much more customized. And they're going to be much more customized because we will be able to develop these digital twin optomechanical models of the eye. So we will have these technologies, and again, I took pictures from my own lab where by three-dimensional OCT, quantitative imaging, we can have the geometry and the structures of the cornea and the crystalline lens, also the gradient index of refraction. We can have through second harmonic generation microscopy the collagen organization, so not only the geometry and the macrostructure, but also the microstructure of the ocular components, as well as mechanical maps. And that will allow us to build these eye models that are specific to every patient. And when you do that, you can actually customize the corrections. So you can produce free-form spectacles that will be fully coupled to the patient's optics, maybe also the visual system. When you have the mechanical properties of the eye, you can actually very accurately predict the response of the cornea to, for example, corneal inlays, that they will be fed then to the mechanical response and the optical response of the eye. You can also do that with customized intraocular lenses or contact lenses, because now you have the eye and you can obtain through optical measurements the lens that best fits the patient in combination with their cornea. So I think we will go to customized corrections. And of course, there will be an evolution of technologies that will enable these customized corrections. For example, freeform optics, and this is work by our colleague in the University of Rochester, Yannick Roland, that will allow to produce any type of surface, not limited to any type of symmetry. Also, meta lenses that will be able to produce very compact optical instruments. This is work by MIT, where they're doing meta lenses that can actually change focus by altering the material with a changing temperature. I also think there will be a revolution of 3D printing. Now there's work showing the first optical surface quality of 3D printing. That's probably a revolution for optics and optical aids in the eye. We have some of that now with light adjustable lenses. So these are intraocular lenses with a material that is cured by UV light changing its shape in vivo once the intraocular lens is implanted. Of course, we have the laser-induced refractive index change. This is work by Wayne Knox at the University of Rochester that can change the index of refraction, spatially resolved across the cornea or intraocular lenses or contact lenses, so that you can produce any custom device in combination or to implant in the eye. So that will leave us, I think, to a revolution in these optical devices that will also make them functional and multi-purpose. Now we have more compact devices. The smaller spectacles, this is something that is already in the market, I think, being assistive, but I think they will also have some type of wavefront correction. They can be deformable, and they will be AI powered. As far as contact lenses and intraocular lenses, I think we're also going to see these multi-purpose devices where the optics will be combined with flexible electronics that can enable sensors, integrated photonics, and drug delivery. So there will be these devices that are not only optics, but that they will encompass electronics and photonics. Actually, I think something that will enable those technologies are biomaterials. I just want to bring here some work that we're doing, which I think is, it has a great future, although it's an old material. And this is a material, biomaterials based on silk fibrine. The silk was, of course, produced for the first time 5,000 years ago in China and was brought to Europe by Marco Polo in the 1200s. But what we've seen is that you can actually do a bunch of things in optics and in visual aids based on silk fibrine. For example, we're doing corneal bandages that can release growth factors, kind of a surrogate of what the enamel membrane does for corneal wound healing. We have also shown that they can act as a really phenomenal substrate for cells and create scaffolds, or they can work very nicely as corneal inlays, doing additive instead of subtractive surgery. So, and those is enabled because it's a transparent material. You can fine tune the biodegradability from days or even hours to actually permanent residency in the eye, and also because you can fine tune the mechanical properties. But what is interesting is that it combines really well with electronics and photonics. So you can use silk-based flexible electronics for drug delivery, for actually printing waveguides. So it's actually mind-blowing how you can bring all this to an old material, but that will lead to these functionalized biomaterials that I think will be key in the next generation of visual aids. So presbyopia, well, I hope 100 years from now, we will have accommodating IOLs and restoration. That can come in several different ways. I'm showing work for my own lab, the light lens, which is an artificial bio-inspired accommodating IOL that is coupled to the accommodative plant. But because we may have all these additional aids combined with electronics, this can be a biomaterial electronic device that is powered from external sources. But also, of course, there could be pharmacological restoration or reprogramming of the crystalline lens aging process. As for myopia, I hope that 100 years from now, we will have elucidated the mechanisms of myopia development, and that there will be effective methods of myopia control. We don't know if this can be from optical means like current contact lenses or scleral treatments or just promoting outdoor activities or any other that will be discovered. In doing some research for this talk, I found that in 1944, the myopia control project was actually by Baltimore myopia control project was launched. We have initiatives now. Hopefully 100 years from now, this will be a solved problem. Of course, I mean, we have been talking about optics, but there will be other critical advances that we are seeing now. Regenerative medicine, stem cells, tissue engineering, gene therapy or editing, and optogenetics to control neural circuits with light and vision restoration. And I think at the end, we will have vision restoration and augmentation. It's probably going to be kind of a blend of biological approaches and technology. Probably not started looking that much as those a little bit creepy AI generated images of how the world is going to be in 100 years, but probably the concepts are there. I mean, this mix of biology and technology. So the vision of the world was not very favorable, I think. But I think with new vision and new vision technologies, we will still be able to see the world, the natural world, and also celebrate in person. As today, being aided by technologies like Zoom with people joining remotely, but also to celebrate in person. And I think Gerald will share this kind of humanistic view, right, of technology. And I know Gerald has always embraced new technologies, but he's always enjoyed music, literature, arts, and I hope the world 100 years from now allows whoever is here to do that. So, well, thank you very much. Thank you very much, Gerald, for all the inspiration and for being with us and being that mentor and friend for all of us. Thank you all for having me, and happy 100th year.
Any comments from Gerald? No, okay. So I think you'll agree that we got a couple of very nice speakers in optics and AI from Washington and from Rochester, but you don't have to look too far than Berkeley if you want to find somebody that is an expert on brain imaging, and Jack, his field hits the nail right on the head. So let me tell you a little bit about Jack Gallant. He's a member of the Department of Neuroscience, holds the Class of 1940 chair at the University of California here. Is a courtesy appointment in EECS, or Electrical Engineering and Computer Science, and a member of the program in Vision Science, Bioengineering, and Biophysics. He's the co-director of the Henry H. Wheeler Jr. Brain Imaging Center on campus, senior member of the IEEE, and served as 2022 chair of the IEEE Brain Community. Jack's research focuses on high-resolution functional mapping and quantitative computational modeling of human brain networks. His lab has created the most detailed current functional maps of human brain networks mediating vision, language comprehension, and navigation. And they have used these maps to decode and reconstruct perceptual experiences directly from brain activity. So the real charge was, here we go. Jack will read it to you, but can you read your patient's brain to see how they're doing on an acuity chart? And so, Jack.
Thank you, Russ. I just want to say how honored I am to be speaking here for Gerald's 100th birthday. I was an undergrad here at Berkeley in Steve Palmer's lab. Gerald's name came up every couple weeks. Then I switched I moved to Yale for graduate work, and one of my graduate advisors was Joy Hirsch, who worked on hyperacuity, and Gerald's name came up multiple times every day. And I read all of Gerald's papers. And then at the end of graduate school, I realized I wasn't smart enough to be a psychophysicist, so I switched to neurophysiology. But Gerald had a huge, enormous influence on my development as a scientist, and I really appreciate being able to talk about a topic of interest to Gerald today. So the title of the talk was in Gerald's email about topics he would like to hear about, and he wanted to know if we could use neuroimaging to try to better determine what people were really perceiving rather than just getting a behavioral response. Now, Gerald refers to a 1920 paper by Kohler. I looked up this paper. I think it was a Fettschrift, some sort of manuscript rather than a paper, and it's in German, and I don't read German, so I have no idea what Kohler was actually asking, but hopefully Gerald will be able to tell me later on. So you might ask, why bother doing brain decoding? I should mention up front, actually brain decoding isn't the main thing I do. It's just kind of a side effect of what I do. The main reason we do brain decoding is it's a kind of a handy way to summarize scientific discoveries for people if they don't want to sit through the details of what you actually discovered. It's also potentially useful for creating brain-computer interface devices, although that's a field really in the early stages. And then it has a third use, which was the thing that Gerald was actually asking about, which is, can we use it to diagnose brain disorders or vision disorders? And right now, functional MRI or any other, let's stick with functional MRI. Functional MRI is not used in the clinic for anything other than pre-surgical mapping. And as you'll see, we can do a lot with MRI. It's kind of a shame that it's not used in the clinic, but there are some real hurdles. The first is that the resolution of MRI and the information it can return, although really extraordinary under the right conditions, is hard to do. It's hard to do well and hard to replicate in the clinic when you're using very constrained conditions with minimal time and patients who aren't necessarily very cooperative. So since we're going to be talking about brain decoding, let me first very quickly take you through how a brain decoder works. It's really not that complicated. First, we have a stimulus and some brain activity that we measure in an individual person. If we're using an MRI machine, we might record brain activity in little tiny volumetric pixels called voxels about 2 millimeters on a side. And there will be about 100,000 of these intersecting the gray matter of cortex. And then we're going to take the brain activity elicited by images or a Snellen eye chart or whatever stimulus you may use, and we're going to try to model that brain activity using some sort of encoding model. The encoding model is simply a computational model that reflects the underlying processes that we think are happening in the brain tissue where the voxel lives. So for example, in primary visual cortex, the encoding model might be a pyramid consisting of Gabor wavelets. We're going to build a different encoding model for every single voxel. And then in the decoding phase, shown here at the upper right, we're going to show the person a new stimulus they hadn't seen before. And we're going to measure some brain activity, and we're going to try to reconstruct the picture that they saw. And the way we do that is we simply invert the encoding models that we obtained from each of the voxels, sum them together, and we create essentially a posterior distribution that will probably be a pretty bad reflection of what they saw. So almost always in these kinds of decoding procedures, you need to include some sort of prior. And the prior is very, very important. First of all, it helps constrain the possible solutions so that, for example, if you know that you showed the person a natural image, you would make sure that the prior only included natural images and not just random white noise. And also the prior helps leverage the data, and essentially it augments the data. So if you have a fairly small data set, as we oftentimes have in human neuroimaging, you can basically get a better answer than you essentially deserve to have with that data set because you have a strong prior. So this is the first natural images decoding result that was obtained from my lab about 15 years ago. You can see that on the top we have the images that were shown to people in the MRI machine, and on the bottom the decoded images, and these aren't bad decoding results. The contrast is wrong because we lose contrast. MRI is measuring blood flow, not neural activity. The neural activity is essentially rectified in the signals we measure, so we don't have black and white contrast anymore. We essentially have a texture map. You can see that the patterns shown here are not, crazy. They're not completely something that's completely randomly related to the images. It turns out you can also decode movies. In this slide deck, I don't have the movies here to show you, but on the left is the decoded results, a static frame from a movie that's being decoded. In the upper left is a person that the subject saw in the MRI machine. In the upper right, on the left 4 quadrants there, the left 4 pictures, is the picture that we're decoding. You can see that this MRI structural model does a pretty good job of decoding the edges in the image. And on the right, we're decoding the semantic content of the image from a higher order brain area that reflects the contents of the images rather than the structure. So this worked fairly well. It's actually kind of surprising that you can decode motion at all, given that MRI signals are measuring blood flow, and there's not really any information in MRI at a time scale faster than about 1 second. But because movies have very long temporal correlations, even if you only have a brain measurement every second, you can decode a lot of information in movies that is correlated with that time scale. Now, the examples that I showed you just prior were taken from a system that used an explicit model. There was an explicit computational model that was essentially designed by hand, a structural model or a semantic model. Different models were designed by hand for different brain areas. And then the prior was essentially just a large sample of natural images or movies that you had to select from. They didn't include the movies or images that were shown, and you had to select from those in order to do the reconstruction. But now with artificial neural networks, we don't have to do either one of those processes anymore. We can replace the explicit feature space with an artificial neural network that has been trained to, say, do image classification, any kind of convolutional neural network. And that convolutional neural network provides features that reflect the likely statistical information in natural images, and you can use the features provided by the artificial neural network to build encoding models for each voxel. The artificial neural network also provides a prior distribution in the decoder. So you can think of a neural network as learning a distribution over all natural images. And because it's learned the distribution of natural images, it essentially embodies a prior itself. And so you can use the neural network both to fit an encoding model to the voxels and to improve the decoded results as you decode them. And so over the last 5 to 8 years, there have been literally dozens of papers that have done this. This is just one that I'm familiar with because it's my former postdoc who did it. On the left, you can see is the ground truth images. And on the far right, you can see the decoded images where he's using 3 different kinds of information. He's decoding text annotations of the images that were actually annotated by a CNN, so they don't come from the subject. They're decoding the structural information in the image using a GAN, and they're decoding the depth of the image using a neural network that was trained to decode depth from 2D images. When you combine all 3 of these kinds of information together, these are all going to be reflected in different brain areas. You can see that it does a pretty good job of decoding natural images. There have been a lot of these papers that have come out. They all perform similarly, and the reason they perform similarly is that our limitation now is no longer our ability to build the decoder. It's the limitation has to do with the brain measurements itself. I should mention that you can also decode images. Excuse me, you can decode visual imagery. But your decoder for visual imagery is not as good as the decoder for perception. And the reason for this is simply that we don't have ground truth for imagery. So to build a perception decoder, we can show you images and actually get a valid model of how you are seeing those images. But when we ask you to image, we don't really know what you're imaging. And so when we try to build the decoder, it's never going to be the right decoder. So there are 4 factors that limit our ability to build a decoder. The quality of the brain measurements, the features that we're decoding, the accuracy of the encoding and decoding models, and the size of the prior and the computer power that we have. And the only one of these that matters today is the first one, the brain measurements. And to give you an analogy, I just want to point out this photograph at the lower right. This is the very first photograph ever taken by Joseph Nucci in the south of France in the early 19th century. This is a really bad photograph, but you can see what it is. And I would say our ability to decode brain activity right now is basically at the level of this photograph. And here we are 200 years later, and now, photography is so good and so efficient that no one even wants to pay for a camera anymore. They just expect to get a camera for free on their phone. And eventually, probably brain decoding will also be at that level. We just don't know how long it's going to take to get there because we don't know what the devices are going to be that are going to allow us to improve our brain measurements. I cannot move forward. Okay. That's okay. So I just want to point out that if we're going to build a brain decoder, the question is, what are we decoding? And there's been a lot of controversy about claims about what is being decoded and what is not being decoded in various studies that have been published in the past. On the left here is a great study from 2005 by Frank Tong, where he decoded orientation. This was the first interesting brain decoding result that wasn't just a binary classifier. And it was thought at that time that they were decoding orientation columns. And later on, Jeremy Freeman and David Heeger showed that actually there's a large orientation bias. This is actually a map of the retinotopic orientation bias. And that large orientation bias allows you to decode orientation from images, even if you have very little information in single voxels that allows you to decode. And there's been a long controversy ever since for the past 20 years about how much of our ability to decode orientation is due to individual voxels containing orientation information versus the entire retinotopic map containing orientation information. And the answer, as always happens in these kinds of things, is it's sort of both. I have lost my capacity to advance. Thank you. So it should be clear, though, to any neuroscientist that what we really want to be able to do is decode columns and layers, right? We want to decode the smallest quantity possible. If we were real neurophysiologists, we could decode individual neurons. But because we're using a non-invasive device, we're not going to be able to get that far. The question is, how far can we go? And if we want to decode an ocular dominance stripe, say in primary visual cortex, we're going to need about 750, 800 micron resolution to do that. If we want to decode an orientation column, nobody's exactly sure, I think, maybe Jonathan knows how big those are in humans, but I don't. I think they're probably something on the order of larger than 50 and smaller than 500, so say 200-300 microns in width. We're going to need a very good MRI device. And people have, of course, pushed really hard to try to decode this kind of information in the brain. Next slide, please. So one thing we can certainly decode is thin stripes and thick stripes in area V2. And those stripes are a millimeter, maybe a millimeter and a half in width. So they're fairly easy to decode using fairly straightforward, simple luminance versus color contrast method. Next slide. A much more challenging thing is to decode ocular dominance in V1. Jonathan had a very nice, and just see you sitting here, Jonathan, I don't mean to keep bugging you, but Jonathan had a really nice paper many years ago that showed that the ocular dominance columns in humans are about 800 microns across, and that's pretty much at the limit of current fMRI. But at 7T, Essa Yakub and Camille Ugabel showed over 10 years ago that you can decode ocular dominance columns. On the left three columns, the left three pictures here are three different runs of trying to decode ocular dominance columns. And the fourth column is averaging the first two runs, and the fifth column is averaging all three runs. You can see that the third run here is quite a bit noisier than the first two. But this does look somewhat consistent. It looks like you can decode ocular dominance columns, and that makes sense because those are on the order of a voxel size that we can measure by 800 microns. Next slide, please. But what we would really like to measure is orientation columns, and I think we don't have any evidence that anybody can measure those yet. So the only paper that I know of that argued that we can measure orientation columns was also from Essa Yakub and Camille Ugabel, and they tried their best to decode orientation in a paper from 2008. You can see there's three columns here: day one clockwise, day one counterclockwise, and day two counterclockwise. And if you just compare day one clockwise to day two counterclockwise, day two clockwise, this should be a pure replication of orientation column decoding. You can see that these maps bear no relationship to one another. And I always get very worried when people try to decode orientation, because those of you who are as old as I am remember back in the 1990s when there was a huge controversy about intrinsic optical imaging and orientation columns. And one of the things that came out of that was the understanding that if you just bandpass filter white noise, you get something that looks like ocular dominance columns. So what you need to do in order to show that you get ocular dominance columns is replicate from day to day to day. And no one's been able to do that yet. That's where we're trying to go. Next slide, please. So our effort here at Berkeley to try to get at this uses a device developed by David Feinberg and a large consortium, the NextGen 7T MRI machine, which produces the highest resolution functional images that a normal, working neuroscientist can actually get. This is a normal 7T magnet, but the guts of the magnet have been completely ripped out and replaced. All the electronics have been replaced with bespoke electronics designed specifically to run specific pulse sequences that can measure the individual capillaries in the cortical columns and primary visual cortex. And this should be able to get us down to a resolution of about 0.4 millimeters functional imaging. Crew in my lab, Matteo Visconti de Elegio Castello, Jen Holmberg, Joseph Vu from UCSF, and Alex Beckett from David Feinberg's lab have worked very hard for the last 6 months to try to get orientation columns in V1, and I was really hoping I would be able to show you some of those today, but I am not satisfied that we actually have it yet, and I don't want to just be another person showing you noise, so I'm not going to show you those columns. But we're working very hard, and I am still quite optimistic that we're going to have the resolution to do this once we develop the preprocessing pipeline more thoroughly. Next slide, please. That's about it. So to answer Gerald's question, I think MRI-based brain decoding at very high resolution is within reach. Once we can decode cortical columns from V1, we can decode columns from everywhere, right? V1 is just the proof of concept because we know what we should be able to decode there. But moving this into the clinic would require getting off the MRI crack pipe and sort of developing a more portable kind of technology that could be deployed widely. And that is much farther in the future. MRI produces hundreds of times more information per time money graduate student than any other method of non-invasive brain measurement that we have. And there is no non-invasive method on the horizon that's going to even come close to it. Anyway, thank you very much. Gerald, thanks so much for giving me a chance to talk.
All right. Thank you very much. The speakers really read the memo. I said, this is not your day. You're just the entertainment. It was excellent entertainment, I have to say. I said, but you're not allowed to go over a specified amount of time. And we are like well ahead of schedule. But that's super important because why are we here? We're here to mix with each other, celebrate Gerald. And so now until about 6 o'clock, there's a bar outside, there's snacks at the back. Soon there will be people walking around with plates of hors d'oeuvres and food. The sun has come out, so they're going to open the doors and we'll just mix and walk around. Somebody will tap the mic. There'll be a few toasts toward the end of the event. So someone will tap the mic, so be sure to come in to hear that. But otherwise, enjoy the next hour and a half mixing with friends.